Hey bio students, this is Mr. Wolf, and in this video I'm going to talk about how to set up dihybrid Punnett square problems. In our last video on monohybrid crosses, I talked about two of Mendel's important laws, the law of dominance and the law of segregation. The law of dominance taught us that when you have a heterozygous individual, such as big P, little p, in pea plants, the big P, the dominant gene for purple flowers, is going to mask the white gene for white flowers in those heterozygous individuals. The second law, the law of segregation, told us that when parents pass on their genes to their offspring, they don't pass on both alleles, both copies of those genes to their kids. Those genes, those chromosomes, go through meiosis and get segregated or separated into individual gametes, which means parents can only pass one or the other allele, never both. Now Mendel's third law is called the law of independent orientation, or law of independent assortment, and this deals with two or more genes found on different chromosomes. So let's take the genotype big P, little p for purple flowers, but also big Y, little y for yellow seeds. This individual is heterozygous for both, but they carry all four possible genes. A purple gene for purple flowers, a white gene for white flowers, lowercase p, a big Y for yellow seeds, and a little y for green seeds. So as this individual goes through meiosis, these chromosomes are separated in meiosis 1, so the homologous pairs are separated. On the left we have big P's and big Y's, on the right we have little p's and little y's. And then we go through our second stage of meiosis where they're separated into individual gamete cells, and we have two possible combinations. We have a big P, big Y combination where we have a purple allele and a yellow allele, a little p, little y combination, a white flower allele, and a green seed allele. But let's say those chromosomes lined up differently during meiosis. Let's say the top chromosomes actually switched places, and this was the orientation they had before they went through meiosis. Going through the steps of meiosis, we'd separate the homologous chromosomes into individual cells. Those chromosomes would then be separated into their single-stranded chromosomes through meiosis 2. But now we have two new combinations. On the left, we have little p, big Y. These gametes carry a white flower gene and a yellow seed gene. On the right, we have big P, little y. These gametes carry a purple flower gene and a green seed gene. So we can see, because of the way those chromosomes lined up, we can have four different possible combinations in the gametes from this parent. So let's apply this to dihybrid crosses. Let's take the genotype big P, little p, big Y, little y. How do we figure out the four possible combinations in a relatively efficient and easy way? Well, we're going to borrow something from math class called the FOIL method. The FOIL method stands for first, outer, inner, last. And to use the FOIL method, I'm going to start filling out our 4x4 Punnett square because we're going to end up with four different possible combinations from this one genotype. Let's take a look at the F. The F stands for the first. So we take the first P and the first Y in our genotype. This happens to be both big letters, so we have big P, big Y as our first possible combination. Now let's move on to the O. This will stand for outer. We take the outer P and the outer Y, big P, little y. This is our second possible combination. The I stands for inner. This will take the little p and the big Y, the two inner letters of our genotype. That will be our third combination. And finally the L stands for last, which will be our little p, little y, our last and final combination. So we have four possible gene combinations for four possible gametes given by this one parent. So let's say we cross two heterozygous individuals that are heterozygous for both flower color and seed color. We can make the assumption that the parent on the left-hand side of our Punnett square will be the same as the gametes at the top because they have the same genotype. So now all we have to do is fill in our Punnett square. So as we go through and fill in our Punnett square, we'll notice that we're always going to keep the letter order consistent. We'll keep the P's in front of the Y's. Even if the P is lowercase, as in this genotype right here, big P, little p, big Y, little y. This is telling me that this possible offspring will have purple flowers and yellow seeds. So filling in all of these, we will see that there are actually 9 individuals out of the total 16 that will have both dominant purple flowers and dominant yellow seeds as their phenotype. 3 of these individuals will be purple flowered but produce green seeds. They are dominant for the first trait, yet recessive for the second one. These 3 individuals will be the reverse. They'll be recessive for the first trait, they'll have white flowers, but be dominant for the second trait with yellow seeds. And finally we have one left, and you guessed it, these are going to be recessive for both. They'll have white flowers and green seeds. So when you fill out this 4x4 Punnett square, you're never going to have to fill out all 16 boxes, because if you can remember 9, 3, 3, 1, 9 dominant for both traits, 3 dominant for the first, recessive for the second, 3 recessive for the first, dominant for the second, and 1 recessive for both, you'll never have to fill out all 16 boxes. But dihybrid crosses are important to be able to do. So let's take this example. We have a heterozygous individual crossed with a homozygous dominant individual for both traits. Well, we've already done these 4 possible combinations for the heterozygous individual. And lucky for us, the second individual has only one possible gamete combination, big P's and big Y's. So when we fill out these boxes, we have a lot less work to do. And that's dihybrid crosses. Feel free to rewatch this video if you need extra help. See you in class.